Welcome back to Dr. Liu's VCE Biology. Today's topic is genetic changes in a population over time, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Describe the functional difference between a structural gene and a regulatory gene. While a structural gene codes for a protein that serves a structural or functional role in the organism, a regulatory gene controls or modulates the expression of other genes. The Hox genes are master regulatory genes that influence cells in a particular location of an animal embryo in order to develop structures for that part of the body. In the brine shrimp, Artemia, the expression of the Hox genes UBX and SCR results in the growth of either a swimming appendage or a feeding appendage, depending on whether the genes are expressed in cells that are in the midregion of the body or that are near the mouth. These specialized appendages are labeled the diagram below. The creation of different appendages in specific locations in the Artemia embryo, despite the involvement of the same downstream genes, is controlled by the expression of Hox genes through multiple mechanisms. Temporal regulation. Genes are expressed for different lengths of time at each location, possibly through varying regulatory sequences that dictate different on and off times. A shorter limb might be the result of a gene switching off earlier or turning on later. Post-transcriptional variation. The resulting regulatory proteins may differ at each location due to post-transcriptional modification or alternative splicing, differential cutting of introns. The BMP4 gene is a key example of how a single developmental signaling pathway can generate evolutionary diversity in different organisms. BMP4 regulates beak formation in Galapagos finches, where its duration of expression in the embryo dictates the final beak size and shape. Parallel to this, the same gene governs jaw formation in African cichlid fish, with its overexpression able to dramatically modify the jaw shape. In both finches and cichlids, this genetic mechanism provides the distinct, naturally selectable phenotypes that contribute to population diversity. Core Concepts Question 6a. Structural versus regulatory genes. Structural genes code for proteins that form the structure or function of an organism, the building blocks. Regulatory genes control the expression, on or off time, amount, of other genes, the controllers. Question 6b. Hox gene mechanism. Hox genes regulate the same set of downstream genes to create different appendages, the structures in specific body locations. This variation is achieved through differences in timing, when genes turn on and off, and post-transcriptional modification, leading to different regulatory proteins. Evolutionary parallel. The BMP4 gene. Question 6c. The BMP4 gene demonstrates how a single signaling pathway drives evolutionary diversity by controlling development in different ways. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.